So once we have an mRNA, it gets regulated as well, and we call this post-transcriptional gene regulation. So what happens to mRNAs? How are they processed, and how can this control expression? So we'll talk about splicing and then stability of mRNAs. Okay, so remember, when we get our pre-mRNA, we have introns and exons. Which piece of that needs to be removed? The introns are the bit that needs to be removed, and the exons get spliced together to create the mature mRNA. Now remember, there were alternate ways we could splice these things. So here there's 2 alternate splices. We have 1, 2, and 3, leaving out 4. And here we have 1, 3, and 4, leaving out 2. And these will give us slightly different proteins. Okay, why is that interesting? Well, here's an example. We have a gene that is involved in flowering in plants here. It's called FLM. The cool thing about FLM is that it's actually temperature regulated. Now, let me walk you through the diagram here, and then we'll talk about the specifics. So anytime you see an arrow in a genetic diagram, that means something's being pushed forward. Anytime you see a bar like this, it means something's being inhibited. So here we have a temperature-sensitive splicing that's occurring. Under low temperatures, FLM gets spliced into the beta form. Okay. Under high temperatures, it gets spliced into the delta form. The beta form of FLM inhibits floral activation. Now, that makes a lot of sense, right? Under low temperatures, you don't want to be producing flowers. Maybe it's winter and there's no pollinators around. Very wasteful to make your flowers. Under higher ambient temperatures, though, it gets spliced into the delta version and pushes flowering forward, leading to flowers occurring. So RNA splicing here is impacting the phenotype that we're getting. And we've even seen this throughout evolutionary time. So here I have 2 flowers. This one has big petals, and this is an outbreeding plant. Pollinators come in, they take pollen from it, and they go to another flower and spread the pollen around. This one is tiny and small, and it relies on selfing. It does sexual reproduction with itself. We find that the cytochrome oxidase protein here, when it's inefficiently spliced, it actually leads to not a lot of this plant hormone called brassinosteroid being made. And that leads to petal cell proliferation. The cells get big and they divide, leading to large petals here. In the other species here, we see very efficient splicing of the mRNA, and that leads to a large amount of brassinosteroid being produced, which inhibits petal cell proliferation. The petals end up much smaller, leading to this selfing phenotype. It's not just plants that this is limited to. We also see this in fish. Here's a marine stickleback. It's got these long spines on it. And here's a freshwater stickleback. It's got tiny little spines on it. In the marine stickleback, the intron is spliced out properly. In the freshwater stickleback, the intron is spliced out improperly, leading to part of the exon being removed. Here, we see these tiny spines as a result. So evolutionarily, why might this be important? Well, if you live in a marine environment, let's hypothesize. Maybe there's big predators out there that want to eat you. And maybe having these long spines discourages those predators from eating you. In a marine environment, that's great. In a freshwater environment, one, maybe there's not those big predators, so you don't need the spines, you don't need to waste energy making them. Or 2, I don't know, here's another hypothesis. Maybe you live in like the shallow areas where there's lots of plants and things like that. Having these big long spines sticking out, maybe you'd get caught and hung up on all the little freshwater plants that are living in there. So there's various hypotheses here, but we see that differences in splicing are leading to different phenotypes that are occurring. All right, so also another aspect of this is not just splicing, but is the mRNA continuing to exist? We call this mRNA stability. If your mRNA gets degraded, it can't be made into protein. Now, it might seem strange to make an mRNA and then degrade it right away, but as long as it works, as long as the organism doesn't go extinct, that's a form of regulation. So basically, changing the stability or how long the mRNA exists changes how much protein gets made. The 5' cap and the 3' poly A tail that we've talked about previously, they protect the mRNA from degradation. So they can influence how long the mRNA exists and then how much protein gets made. There's some correlation between the length of the poly A tail and how long the mRNA stays around. Okay, there's also ways of also influencing mRNAs through other means. So these are called microRNAs. MicroRNAs are a short non-coding RNA that can bind to mRNA. So here's the structure of a microRNA. It makes a hairpin loop structure, and there is part of the microRNA that is complementary to the mRNA. So that means it can base pair with it. When the microRNA piece binds to the mRNA, there's two different things that could happen. In one instance, it might bind and lead to degradation of the mRNA. That degradation is going to destroy the mRNA, preventing it from becoming protein. In the other example, the microRNA might bind to the mRNA and block translation. So I want you to think about this. How could a microRNA interfere with translation? I'll give you the first one, right? If it leads to degradation and chopping up the mRNA, that's going to inhibit translation for sure. But think about the second example where it binds to it. Now remember, the process of translation involves the ribosome coming down and scanning along that mRNA. If you have two pieces of RNA bound together, that's going to block that ribosome from translating. So that's a way that the second mechanism can block translation. Again, we have this T-bar here indicating it inhibits translation. Okay, so alternative splicing is one mechanism of post-transcriptional regulation. We can create different functioning proteins here. Changes in the splicing have impacted development and evolution. I showed you a couple different examples. RNA stability impacts if an mRNA becomes a protein or not. So the 5' cap and the 3' poly A tail protect mRNAs. And the longer an mRNA exists, the more protein is going to be made from it. MicroRNAs, on the other hand, are very short RNAs that base pair with mRNA and lead to the destruction of mRNA, or they could block translation of that mRNA. Okay, we're going to keep moving down to the protein level next.